Good morning.
Good morning.
Good morning, everybody. Good to see you. We're just a couple minutes early, so we'll let other people join in. Go ahead and type your first name or your nickname into the chat for me, if you would. All right, very good. So a little bit of housekeeping. You have an exam today. Don't forget about that. I saw this morning that some of you have already taken it. Very good. Get it out of the way. Remember that the lecture exam is open to you today until 8 o'clock, or a little past that, actually. It's due at 8 p.m. So make sure you find a block of time that you can sit down and complete that exam. Remember, you're allowed to use your lecture notes while you take these lecture exams and lab practical exams. You're allowed to use the notes that you make. So, but you're not allowed to use any other materials. You're not allowed to get any assistance from anyone. So keep that in mind. The last time that we were together, I spoke about being sure to keep track of your grades, keep track of what you've handed in and what still needs to be handed in. Make sure you stay on top of that. I know it gets very burdensome when you're taking multiple courses, especially multiple online courses, to keep all of your due dates in track. Do whatever you have to do to check and double check on yourself to make sure that you're getting your work in on time. Remember that in this course, at least, you are allowed to hand in late work up to 1 week past the due date. So if you can't get an assignment in for whatever reason, you are certainly welcome to submit it late. You may lose some points for the late submission, but it's better than not doing it at all. And of course, if you ever have something come up in your personal life that requires you to attend to it and not get an assignment in, just message me, let me know. And if you ask for an extension on something, I'm happy to work with you that way. Several of you have had just things come up and you've needed a couple days extension on work, and that's fine. I absolutely recognize that things happen in our lives, and sometimes we just need a day or two to get caught up on things. So don't forget that. Today, we are going to be working on one of our 2 lab topics for this week. Today, we're going to be talking about microbes in the environment and how we work to monitor those microbes and not only find out if they're there, but if they are, to identify them, to sample these environments and identify these microbes. We'll start that today. We probably won't finish it today. We'll finish it on Wednesday. And then on Wednesday, we'll also start talking about what's called special media. And these are types of culture media that allow us to really differentiate between organisms and be able to identify certain groups of organisms and even certain species of organisms, depending upon how they grow on these particular kinds of culture media. So those are the 2 things that we have coming up this week. I'm going to go ahead and pull up our Blackboard page so we can all take a look at our to-do list for the week. You can see at the top of the to-do list is the lecture exam. And of course, that's today, March 1st. I can't believe it's already March. Um, you have one lecture topic this week, so one set of lecture videos to watch, and those are about microbial biochemistry. Now, I did want to say just a word about this lecture. I think this is one of the most difficult topics that we cover in this course. When I use that language, biochemistry, I'm literally talking about the chemistry that applies to biological things, to living things. So it's a lecture that covers the kinds of chemistry that we need to understand as we study microbes. And I recognize that many of us don't enjoy chemistry as much as we might enjoy other subjects. It can be quite difficult at times, chemistry. So make sure you give this lecture topic the time it deserves. You may need to give it a little more time than you're used to compared to other lecture topics we've had so far. So just be prepared for that. And of course, there's a quiz associated with that lecture that's due Friday by midnight. And you'll have lab homework, as usual, due by Sunday at midnight. And of course, all of those materials that you're going to need during the week are here for you as well. So be sure if you've just come in to type your name or your nickname into chat for me. So I have a record that you're here. Thank you. It looks like just about everybody's here, which is great. I'm going to go ahead and begin our lab material. But first, before I do that, is there any questions you'd like me to address? If there are, you can type your question into the chat. If you have any questions about the lecture exam today or questions about any of the other materials that we've looked at, that we've talked about in lab, anything at all. All right, and if anything comes to mind, you can always type it into the chat, even during the lab, and I'll get to it. Okay. All right, so I'm going to pull up our lab slides. And again, this week we're going to start by talking about microbes in the environment. So this is sort of a special area of microbiology, this monitoring that takes place of microbes around us in various environments. Now, our objectives for this exercise today are first to talk about the kind of testing that we do and the kind of methods that we use when we want to take a sample from various environmental locations in order to see if there are microbes there and to figure out what kind of microbes are there. We're going to talk also today a little bit about culture media. Remember, I said that our second lab topic this week is about this type of culture media called special media. So today we're going to talk more about some basic media. We're going to talk about this Auger material that we use for solid culture media. We're going to talk about why it's such a nice choice for growing microbes on this solid surface. We'll look at the way we use Auger. We'll compare these Agar plates that we've talked about with two other Auger culture containers, some that are called Auger slants and some that are called Auger deeps. And we'll talk about the way that we sort of encourage bacterial cells to grow on these solid media and also in liquid media. The techniques that we use when we spread cells onto an Agar plate are going to differ depending on what we want to achieve, how we want those cells to grow. The last material in this particular lab is about something called colony morphology. Remember, this term morphology refers to body form. So when you talk about the morphology of an organism, you're talking about what it looks like. Now, specifically, this is colony morphology. So we're going to talk about how we use the appearance of bacterial colonies to learn something about the organism. Because it turns out that bacteria grow into these collections of cells, these colonies, in ways that characterize the organism. So colonies have different sizes and different shapes and even different colors. And that appearance can actually help us identify the organism sometimes. So we'll talk about that towards the end of the material. Remember, it's important for us to remind ourselves that microbes are everywhere, literally everywhere. They are on and in all living organisms. Remember, you and I have a microbiome, but so do every other living thing. Plants are covered in microorganisms. Again, those are their own microbiome. Other animals have their own microbiome. So we are not unique in that. Microbes are all over and within at least the larger forms of life on this planet. But microbes also inhabit all different kinds of natural environments. So whether you're talking about soil or you're talking about bodies of water, there are going to be microbes present. We also find microbes in man-made environments. And of course, the majority of those microbes are not harmful to us. We've talked about that. But there are some environments, some man-made environments. So think about buildings and facilities and so on. There are some man-made environments where we can't tolerate even harmless microbes growing. So we need to monitor to make sure they're not present. And if they are present, we need to identify them. Most microbes are harmless to us and even beneficial to us, as we've talked about. But there are a few pathogens out there, and there are times when we have to be very careful that we don't allow them to grow in our environments. So we routinely monitor different types of environments in order to keep people safe, basically. Recreational bodies of water are one of the example environments, especially in a place like New Hampshire, where we rely so heavily on tourism for our economy. We have many, many, many lakes and streams and rivers, and we have a lovely coastline. And we want to keep those places safe for recreational use. Our water treatment plants have to be functional. They have to do the job of filtering and treating our drinking water to keep it safe. We have all kinds of municipal water systems in our country. So the systems that deliver water to people's homes, they have to be monitored. People, again, especially in a place like New Hampshire, many people get their water from wells dug on their land or from cisterns, above ground cisterns that hold water. Those have to be monitored. Of course, our hospitals have to be monitored. And unfortunately, hospitals are a place where we often find pathogens growing in the environment. Remember, some of the sickest people go into hospitals. So it makes sense that pathogens get shed and sometimes find places to grow in the hospital building itself. Even doctors' offices have this problem sometimes. They have this problem of pathogenic microbes finding a place to grow in the facility and being therefore dangerous both to patients and to people who work there. Nursing homes are susceptible to this. Rehabilitation facilities are susceptible to this. I mentioned that there are workspaces where we can't tolerate even harmless microbes growing. And those are places like industrial clean rooms. Think about the factories where things are built and not even a speck of dust can be allowed in those manufacturing rooms because any sort of particulate matter would damage whatever's being built. Think about the rooms where chips are manufactured for all of our devices. Think about pharmaceutical manufacturing facilities. They can't tolerate any microbes growing, so they have to constantly monitor. Now, there are lots and lots of ways to take a sample of an environment. And of course, it's going to depend on the type of environment. In places like hospitals and cafeterias and nursing homes, places where we're concerned about pathogens, we're often sampling solid surfaces. And the best and easiest way to do that is with a swab. In this first image here at the top, you can see a gloved hand holding what is a commercially produced swab for sampling solid surfaces. So this swab comes in its own sterile tube. You can see that the cap for the tube is attached to the swab. And this swab is designed to be used to literally swab or run this instrument over surfaces. And then you take it and you put it right back into its container. And that container has transport media in it. It has some nutrient culture media in it to help keep any cells alive until they get to the laboratory. So we're using these to see if there's anything living on this surface, and we're grabbing a sample. Then it gets sent to the lab, and once it gets to the lab, it will be used to inoculate some culture media. Sometimes we're not trying to sample surfaces, but we're trying to sample the air. This image right here shows, again, a hand with, I think it's just a Q-tip swab, and a ridiculously dirty ventilation grate. But it does serve to remind us that in a lot of spaces, man-made environments, buildings and homes and so on, we have air circulated through that building through ventilation systems. And it is possible for microbes to get a hold and grow in those duct systems or on these grates. So we have to monitor for that. We have to monitor to see if we have any particularly dangerous organisms growing in our ventilation systems. This image over here, you can see a person using a swab to take a sample from a floor drain. Again, there are some places where we have to be really careful to watch that no pathogens start to grow. Floor drains are very common in places like hospitals because we often are washing down rooms, sometimes with actual hoses, and all of that wash water is going to go down the drains. And this is a place for us to see, were there any pathogens in that wash water? Were there pathogens in this room that got sent down this drain? Let's see what was growing. Let's see if there's anything growing in the drain itself that might accidentally cause problems for us. So we're constantly sampling surfaces as well as air, as well as even things like liquids. You know, you can see someone holding a Q-tip in this image with the air filter, but we could also place a filter, an actual piece of paper filter material into a vent, and we can collect microbes on that filter paper and use that filter paper in the laboratory to inoculate culture media. Sometimes we need to collect a liquid sample because we want to determine if there are any microbes growing in that liquid. Remember, we talked about recreational bodies of water. All of these types of samples have to be done with aseptic technique. You need a sterile swab or a sterile container to collect your sample. And we have to be careful to get that sample to the laboratory quickly so that the cells don't die in transport. So we can grow them and determine what they are. If we're doing the sampling, in other words, we have to be careful that what we're getting onto that swab or into that container is handled correctly. If you're going to use a swab like these commercial swabs here with the different colored handles, these are different sizes of swabs. I know they're hard to see because they're white, but these swabs are used for different purposes, for sampling different surfaces, and they all come with their very own container. And this container has liquid media in it. It has transport media in it, so that if we do pick up some microbe on this swab, when we put it into the transport media to send it to the lab, we'll keep it alive until it gets to the lab. When we sample bodies of water, it's a little more complicated. Probably the hardest thing is that we have to get a representative sample. Now, what do I mean by that? If you look at this picture here in the corner on the left, this is a picture of a couple of technicians, I guess we'll call them, and they are sampling a lake. It's probably this big, enormous lake, and I think you can see there's an area that's been roped off, probably for swimming. What you don't want to do here is take a single sample. You don't want to go out here and just grab one sample of this water because that would not be representative. You're going to need to take several samples. You're going to want to go to the far corners of the swimming area. You're going to want to take a couple samples right at the shoreline of the swimming area. You're going to want to get a couple from the middle area. You're going to want to get enough samples so that you feel sure that you've gotten the whole swimming area sampled. When you're doing lake samples like this, other than getting wet, collecting the samples, it's really not difficult. But there are other bodies of water that we don't routinely go into to get our samples. So sometimes you have to use something like this, which is a telescoping pole. You can just see that this person has a plastic bottle at the end of this pole. And again, they're going to use it to get some samples from around this natural body of water here. We can suspend collection containers from ropes, and we can get samples from wells, for example. We certainly sample at the ocean, again, where people swim primarily. And you just have to be sure that you're collecting a representative sample for the whole area. The containers that we use are generally just inexpensive plastic containers. There's nothing special about these. These don't have to be sterile. These don't have to be sterile. We know that there are microbes growing in natural bodies of water. We know there are. What we're trying to figure out most of the time is whether or not there are any dangerous microbes growing here, any pathogens. Now, of course, when you're sampling water, you're going to get a lot more water in your container than microbes. The number of microbes, even when you have an outbreak, even when you have some pathogen that's growing at high levels in a body of water, the number of cells is going to be very low compared to the volume of water that we collect. So we often have to centrifuge these containers. We have to put them in a centrifuge and spin them so that any and all cells will come down to the bottom of the container so we can examine them. Now, in this slide, you're looking at some specialized devices. There are lots and lots of these on the market. These are designed to sample air. So again, going back to things like ventilation systems and sometimes even residences, residential homes and apartments. What these devices are measuring specifically is spores. So we've talked in here about how there are lots of bacteria that can sporulate when the conditions get stressful for them. Remember, we said the two most common conditions that will cause bacteria to sporulate are desiccation or drying out and a lack of nutrients. When bacteria encounter those conditions, if they can sporulate, they will. They'll go from being vegetative, from a cell, into an inanimate object called a spore. And the nice thing about a spore, if you're a bacteria, is that it can survive in dry conditions. It can survive without nutrients. It's inanimate. It's not metabolizing. It's not in need of anything. It can sit there for long periods of time. And then when the conditions get better and better for growth, it'll go back into its cellular form. Now, we also mentioned in lecture that there are two types of bacterial pathogens that can sporulate. Remember, the genus names are Bacillus and Clostridium. So any bacteria that has the first genus name of Bacillus or the genus name of Clostridium is able to sporulate. We have to remember that because those are some pathogens that can give us a lot of trouble because those spores can last for long periods of time. Remember too, that fungi are also capable of sporulating. But here's an important distinction. Fungal spores are not really survival structures so much as they are reproductive structures. Fungi make spores when they're ready to reproduce. Now, these devices, they count spores. And a real market has come up, for better or for worse, for homes. You can call these companies now and have them come over and measure spores in your house. And a lot of these companies, they'll use these very impressive looking devices and they'll tell you, oh, you have 52,000 spores in your bathroom. And it all sounds scary and it sounds like you need to do something. But here's the important thing for us to know as students of microbiology. This device cannot distinguish between a dangerous spore and a spore that is perfectly harmless to us. It cannot tell you that the spore that it's measuring is a pathogen. And the truth is that our homes are full of spores. And people get a little freaked out when you tell them that. But the truth is there are microbes all around us all the time. And again, most of them are perfectly harmless to us. And we are surrounded by spores in our environment. Homes that are older, homes that are particularly dry, and in New England, that means all homes in the winter, right? They're going to be full of spores. It doesn't mean there's a problem, though. It doesn't mean that there's a problem that a homeowner should be afraid of. So it's unfortunate, but a lot of these companies that come in and measure spores inside of residences don't do any follow-up work to figure out if those spores are really dangerous or not. So something to know about that industry. Now, there is another testing technique that I want us to be familiar with, and that's because this is one that you may encounter in your professional life if you work in a healthcare setting. What you're looking at in this picture, this person with the glove on, is holding what looks like a petri dish, yeah? But this is not a petri dish. This is a particular sampling tool called a Rhodac Plate. That word Rhodac stands for Replicate Organism Detection and Counting. These plates are actually much, much more flexible than a petri dish is. Petri dishes are made out of hard plastic. These are made out of very flexible, soft plastic. Like a petri dish, though, like a petri dish, they have a layer of agar on the bottom, and that's what you're seeing here, this beige color. That's just nutrient agar. So when you buy these Rodak plates, they arrive sterile. You take the cap off, you turn the plate upside down, and you literally push the bottom of the plate sort of outwards so the agar can touch surfaces. They're very flexible. You can push on the bottom of the plate and you will poke out that agar surface so that it can touch these surfaces. If there are any cells on these surfaces, they will stick to the agar and they'll grow there after this plate gets placed in the incubator. So these plates are commercially available. People in hospital settings and healthcare settings, they purchase these and they use them to do their own sampling of the environment. I do want to point out, though, that you could also just use a regular agar plate to do this. You could use a swab, sample a surface, and then spread that swab out on an agar plate. And that happens every day in microbiology labs. The difference with the Rodak plate is that it's so easy. You don't need a swab. You don't need a person who knows how to swab a plate. You just need a person who can take the cover off, flip the plate over, flip it upside down, push out the bottom of the plate, the agar surface, so that it touches the surface, then flip it back over, put the cover on, and send it off to the lab. They're very easy to use. Now, this brings me to an important thing to know about how we streak agar plates, and we haven't really talked about this too much yet. You're looking at two different agar plates here that have bacteria growing on them. This particular plate was made using what's called a quadrant streak method. Quadrant, okay? This one over here looks very different, right? The cells are growing very differently on these two plates. Over here, you can see sort of a smear of these cream-colored organisms all growing together. And then as you come down, you can start to see individual colonies of bacteria growing. So the way you make a quadrant plate, like the name suggests, is by dividing the plate into 4 quadrants and then using a streaking technique to get fewer and fewer cells from one quadrant to the next, to the next, to the next. Now, let me tell you what I mean by that. When you take your loop and you dip it into your broth culture, you're going to go into your first quadrant and you're just going to smear the cells around. That's going to give you this kind of growth right here. Then you go ahead and flame your loop, clean it off, get all those cells off that are left over. You're going to put it on the agar plate and you're going to draw it through the first quadrant into the second quadrant, and you're going to smear those cells out. So what you've done is you've taken a clean loop, and as you draw it across the surface, you're picking up some of these cells, but you're not going to get as many cells as you got in this first collection. You're going to get fewer cells, and you're going to spread them out. Then you're going to take your loop and clean it off, flame it, or incinerate it. You're going to draw it through the second quadrant, pick up a few cells and spread those out. And then you're going to do it a 4th time. Draw your loop through the third quadrant, pick up a few cells and spread them out. So by traveling from quadrant to quadrant to quadrant, you're spreading out fewer and fewer and fewer of the original cells. Now, why would you want to do that? The reason we make a quadrant streak plate is because we're trying to grow colonies. We're trying to grow these little individual circular structures called colonies. Remember, I said a few minutes ago that we can look at colonies. We can look at their morphology, their body form, and we can know some information about the organism. We can't identify it directly from colony morphology, but we can gather some data to help us identify it. So the purpose of creating a quadrant streak plate is to grow individual colonies on that plate. Now, compare that to this plate. This plate is covered in individual colonies, right? In fact, that's the only thing growing on this plate. We don't see any of these big, thick smears of growth. All we see are individual colonies, and they cover the entire plate. This is called a spread plate. We don't use spread plates for colony morphology. Instead, we use spread plates to count colonies so that we can do a little math and figure out how many cells were in our original environmental sample. We're going to talk about how to do this later on in the course. But for now, understand that to get a spread plate like this, what you have to do is get your loop full of cells, just like you did for this. You have to get a loop full of cells from your broth culture. And you have to use a device to spread them evenly all over the plate. Remember, we talked about that hockey stick device when we were looking at aseptic technique. So you use that flattened hockey stick tool and you spread the cells all over the plate. And with any luck, you'll get nice colony growth all over the plate. And you will then set out to count these colonies because that number can be used quantitatively to help you determine how many cells were in your original sample. So, if you're working with, say, surfaces, maybe you're looking to see how many colonies are growing in a hospital room, a patient room, maybe. You can do that per surface area, yeah? If you're looking at a body of water, you can figure out how many cells there are per volume of that water. One thing for us to know today as well is that when we count colonies on an Agar plate, we call them something other than colonies. We call them CFUs. This abbreviation stands for colony forming unit. Colony forming unit. It's another word for a cell. We're saying that a cell landed on that Agar plate and grew into a colony. So it's a colony forming unit. Now, what exactly are these colonies? We know they're bacteria growing, but what exactly are they? What definition do we use for this term colony? What a colony is a visible mass of bacterial cells. So that's important. We can see it with our naked eye. We don't need a microscope. It's a visible mass of bacterial cells, and they are all clones because they all arose from a single cell that landed on the Agar plate at that location. So when I spread my water sample, for example, all over this plate, what I did was I spread individual cells out all over this plate. And then I put the plate in the incubator. And overnight, all of those cells that landed on the Auger divided and divided and divided and divided over those 24 hours, creating a little mound of cells, a little mountain of cells that we call a colony. And remember, bacteria reproduce asexually. So when a bacterial cell divides, it creates an exact copy of itself. So they're clones, they are genetic clones. Each parent cell, as we call them, divides and creates two daughter cells. That's the language we use. And then each one of those divides, and then you get 4 cells, and then you get 8, and then you get 16. And in a 24-hour period, you're going to get many, 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 many copies of that parent cell in that colony. And again, the whole reason we count colonies is because we want to be able to extrapolate that number back to figure out how many cells were in our original sample. Does that make sense? What a colony is? What we see on that plate, it's not one bacterial cell, it's a whole bunch of bacterial cells, many, many thousands of them. Because bacterial cells divide asexually, they also divide very rapidly, and they grow exponentially. So over 24 hours, you do get thousands and thousands of cells growing from every single individual cell that was in your original sample. Remember, we can look at colonies from quadrant street plates, and we can know what the colony morphology is for the organism. We can also use spread plates to count them and to then do some math. Again, we'll talk about that later in the semester. That will help us extrapolate back to be able to say, oh, there were 30 cells per ml of water, or there were 5 cells per surface area in the hospital room. So depending on how we put our cells onto our Auger plates, we use the Auger plates for different purposes. I do want to mention one thing here, not to be confusing, but just so you know. Sometimes students will say, why do we talk about CFUs? Why don't we just call them cells? If we're going to call a cell a colony forming unit, why don't we just call it a cell? The answer is because colony forming units are actually just an estimate of cell number. We are not literally counting all of the cells that we put onto that Auger plate. What we're doing is we're getting an estimate of cell number, so we refer to them as CFUs. Any questions? Any questions about any of this so far? So we can sample any kind of environment, water, air, surfaces. You can sample soil. You can gather up some soil. A lot of people do this in their garden, especially here in New Hampshire, because we have an excellent state-funded laboratory that will help gardeners get their soil to the best conditions for growing a home garden. So you can gather some soil samples. Again, you have to get several to represent your whole garden. And you can send them to the lab, and for a small fee, they'll tell you things like whether or not you have enough nitrogen and whether or not there's enough phosphorus and things like that, as well as how many bacteria there are in your soil. There should be bacteria in your soil, and there should be fungi in your soil. And if there aren't, you may want to consider supplementing with something like compost. So we can sample all kinds of environmental materials to determine what kind of microbes are growing in them, both the kind of microbe and the number of microbes. Now, of course, in a healthcare setting, we have very different objectives In a healthcare setting, we're trying to prevent pathogens from getting established in the environment and growing up to a degree where they could harm people. All right. On this slide, you're looking at, over on the right here, you're looking at an agar plate. It happens to contain what's called blood agar, and you can see it's a spread plate. There's lots of colonies growing across the surface of this plate, all over the plate. I put this up here because I want to say just a couple things about how we count, again, colony forming units, as we call them. You don't have to count every single colony that you see. It's much easier when you count plates by hand to take a Sharpie, and on the bottom of, on the bottom of the plate, you draw a line, a vertical line, and then a horizontal line. Again, you create 4 quadrants. You can count up the colonies in 1 quadrant and then multiply by 4. Again, you're not counting every single colony. It's an estimate, but that's okay. We know it's just an estimate. So that's done a lot. We count a portion of the plate and then multiply it to get the total number of CFUs on the plate. In lots of very busy laboratories, though, people don't count plates by hand. Instead, you can use what's called a semi-automated system, or you can use a fully automated system. What you're looking at down here in this image is a person using one of these semi-automated counting systems. So you can see there's a digital display here. And you can also see that there's a grid over this light source. What you can do is you can put a plate into this little chamber here, and the grid will basically show up through the bottom of the plate because of the light. And then you use this special stylus, and all you have to do is touch the stylus to one of these colonies, and it will get registered here. So instead of you having to keep track, you can literally just tap, 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 tap across all the colonies, and the unit will keep track of the count for you. So this speeds up your work a little bit. Now, this plate is actually being imaged in what we would call a fully automated counting system. This is computerized. You would only find one of these in a very busy microbiology lab that had to count a lot of plates every day. But this is actually an image created on a computer screen. I think you can see the menu over here. So you put this plate underneath a camera, and the software captures an image of the plate, and then the software actually does the counting. The software will count up the colonies on the plate and report it to you. So that's a fully automated system. All you have to do is put the plate under the camera, and the computer does the rest. Now, what about these Rhodac plates? Remember, we said some kind of a technician in the hospital or the doctor's office or wherever is going to collect the sample by pressing the Rhodac plate down onto a surface. And then that Rhodac plate gets sent to the lab for analysis. So what happens in the lab is the plate will be put in the incubator just like any other Agar plate. And then the following day, someone in the laboratory will count colony forming units. Now, the way it works is for Rodak plates only, the count that you get is going to put you into a category of either good, fair, or poor. I'll show you a chart in just a moment to explain this further. But the company that makes these plates publishes basically a chart that will show you, depending on different locations in the hospital, particularly, depending on different locations in the hospital, how many colonies you count will determine how good or poor of a job you're doing disinfecting in the hospital. Now, this particular table that you're looking at here, this is one that comes with the plate. So let's take a look at this. You can see in the first column here, this is just listing the different areas that you might sample in a hospital, right? We have things like floors, and they've got different parts of the hospital separated out here: the operating room, the OB, the obstetrics wing, the nursery. You can imagine these are particularly important places to monitor for the growth of pathogens. Tabletops, you can see patient rooms, you have the floors, tray tables, the bathrooms in the patient rooms, the sink, the tub, the toilet seat, other floors, other surfaces. So you have to sample all through the hospital, and then you compare how many colonies grew on the Rodak plate with this chart to tell you how good or bad a job you're doing with your disinfection program. You can see this second column here tells you what your CFU count would be if you're doing a good job. And then this column shows you a fair job, and this column shows you a poor job. So let's look at one of these. Let's say I did some Rhodac plates in the nursery at the hospital. It's my job to sample in the nursery once a month. And let's say that on that plate, I grew 12, or on those plates, I grew an average of 12 colonies. So according to this, here's that nursery category again. If I found 12 colonies on average per plate, I'm only in the fair category. I want to get down to 0 to 5 colonies per plate to feel like I'm doing a good job. Remember, microbes are everywhere. So even in the best, the cleanest hospital, the most well disinfected hospital, you're gonna find microbes growing. The question is how many? How good a job have you done keeping the number of microbes low? And according to the chart, you can get an idea of how good or bad a job you're doing. Now, I want to mention to you that there are a lot of facilities. Let me go back to this for a sec. There are a lot of facilities that use a much stricter standard than this. In other words, there are hospitals that make up their own charts for this kind of testing. There are hospitals where if you got even 2 or 3 colonies on your plate, they would consider that fair, and they would want, disinfecting to be done and a repeat test to be done. So in other words, this is just the table that the company makes. And there are lots of healthcare facilities that hold themselves to a stricter standard than this. And they want, they want to make sure that their disinfection programs are really, really excellent. So to summarize what we've been saying, we can count colony forming units that come from samples that were grown up in broth, grown up in liquids. We can do that quite accurately using spread plate CFU counts. And again, you have to do a little math here, but we can do it pretty accurately and get a good estimate for how many colonies are in that sample. Or I should say, how many cells are in that sample. We can sample surfaces through all kinds of environments using swabs, using Agar plates like Rhodac plates. And we can then grow up any cells that land on those plates and count colony forming units again. But remember this. If I touch a Rhodac plate to, let's say, a desktop surface in a hospital, and I get a count, a CFU count, and I use it, I compare it to this chart to tell me how good I'm doing. I have to remember that I can only speak about the surface that I sampled. So if I get a Rhodac plate back that I pressed to, let's say, a desk at a nurse's station in a hospital, if I get a count back and it's zero colonies, The only thing I can conclude from that is that that desk is clean. That desk has been well disinfected. I can't say anything about the patient room down the hall or the nursery on the next floor. If I want to evaluate how clean those places are, I have to sample in those places. And I have to take multiple samples so that I have a very good representative idea of how clean those places are. So Rodak plates, we don't just take one sample. We always take many, many, many samples from lots of different locations so that we have as much data as we can generate. Now, there are limitations to this kind of testing. There are limitations. This is not a perfect system. If you think about those Rodak plates, the limitation is that I'm only measuring the cells where I pressed that plate. And you can imagine, while it's easy to press that auger down onto a nice flat solid surface, I can't use a Rodak plate in a tight corner, right? I can't press it into a crevice. I can't get it down into cracks. So I might have to also use swabbing to get into certain areas if I want to find out if there are any microbes growing there. All right. Any questions so far? Any questions about how we gather these samples? Remember, the folks that are sampling in a place like a hospital are typically just employees at the hospital. These are folks that get paid as part of their job to do this periodic sampling. Um, these environmental control programs in hospitals, they take this very seriously and they will pay certain employees to do this sampling for them at certain intervals, like once a week or once a month or once every 3 months. It's an important part of making sure that the number of pathogens that might be growing in the environment of the hospital is kept to a very low level. All of those samples that get collected, you can't just stop with the sample. You have to do something with the sample. You have to either inoculate broth with it or you have to put it onto a plate. And those then have to go into the incubator because we're trying to get a number, we're trying to get a quantity. And the best way to do that is to do these colony forming unit counts. So that's how that's done. That's done in the laboratory. We're going to switch gears here and spend a couple minutes talking about culture medium. Remember, we're going to be talking about very specialized types of culture media in this course, and we're starting that this week. So what I want to do today in the time we have left is talk about some general types of culture media. Remember that if we're going to grow a microbe in the laboratory, we have to give it something. If we pick up cells out of the environment, if we take a swab and we swab a drain in a sink in a cafeteria, and we happen to pick up some microbial cells on that swab, we need to grow them. We need them to divide and increase in number so we can find them, in other words. And in order to do that, we have to give them nutrients. We have to give them some media in order to grow, in order to divide. Lucky for us, bacteria and other kinds of microbes have a very short list of nutrients that they require. They've got to have some water. All living things require water. They have to have a carbon source. They have to have a molecule that they can essentially eat that will give them carbon to build other molecules with. Now, that's generally protein for a microbe. protein, and you'll see this word peptone. You'll hear me use this term a lot this semester. All a peptone is a protein that has been partially digested. So what we do in the laboratory is we take protein sources and we add enzymes in that start to break them down so that they're not quite so big and enormous. They're little shorter bits of protein. If you're familiar at all with baby formula, you can buy baby formula that has been partially digested in terms of the proteins. Some babies just can't break down normal protein sources. They need them to be they need the digestion process to have already been started. And it's true with bacteria too. Some bacteria need us to do the first part of breaking the protein down for them. And that creates, again, what we call peptones. So you've got to have a protein source or a peptone source in there. You also need an energy source, and this is carbohydrate. Just like you and I, bacteria and other microbes need carbohydrate to make ATP from. And finally, you need several microminerals. So you need things like nitrogen and phosphorus and sulfur and so on. So our media that we can make in the laboratory, we can design them, although nowadays there are plenty of them already available commercially. They have to have these minimal things in them in order to work, in order to keep cells alive and let them grow. Now, when we have a basic, simple list of ingredients in our media, we give that a name. We call that a chemically defined medium. This is a medium that has known ingredients in it. Everything that's inside of this medium, we know. We put it in, we know exactly what it is. It's chemically defined. And lots of bacteria will grow in chemically defined media. But there are some microbes that are really hard to grow because they have very particular nutritional requirements. So they just can't make it on this basic sort of media. So what we do for those organisms is we add in ingredients that are a little bit mysterious in terms of their chemical composition. We can add in things like blood. We can add in eggs or milk. We can add in extracts from certain kinds of meat or fish. Now, obviously, we know if we're putting blood in, we know if we've added egg into our media. It's just that we do not know every single molecule that is present in blood. We do not know every single molecule that is present in, say, a chicken egg. We know a lot about eggs, right? We know how much protein is in them and how much fat is in them. We know that albumin is in eggs. It's a type of protein. But there are small quantities of all different kinds of things in eggs and meat and blood that bacteria may find very useful. But we just don't know the exact list of everything that's in those food sources. So we don't call those chemically defined. We call those complex media. They're very complex because they have these very complicated natural food sources in them, like blood and eggs and milk and things. If you're curious, the blood that is typically used when we add it to Auger tends to come from sheep. So it gets collected from sheep and then it just gets added into the Auger. So we have lots and lots of chemically defined media available, and we also have lots of complex media available to us. We've talked in this course so far about nutrient broth and nutrient agar. That's one of these chemically defined media. It has a very short list of ingredients in it, and we know exactly what's in it, molecule for molecule. Now, remember, when we make agar, when we make solid media, There is a material called agar. We've talked about this. Agar is a carbohydrate that certain species of algae, these are marine algae, they're called red algae, they use agar as their cell wall material. So we harvest agar from these red algae and we put it into a powderized form and we use it to make our solid media. Now, agar itself is not nutritional, and I'll talk about that in just a sec. Agar is just the sort of the framework for the media. We have to add in the nutrients to the agar. Agar makes a perfect culture medium, a perfect solid culture medium. And I've got some reasons listed here. First of all, agar stays solid in the incubator. That's critical, right? We grow bacteria at warm temperatures, hot temperatures, 35 to 37 degrees centigrade. We can't have a material in our petri dishes that is going to melt in the incubator. That won't work. Agar stays solid in the incubator. You have to get it up to 100 degrees centigrade to melt it. Once it is melted, though, it's great because you can pour it very easily into your containers, like your petri dishes, and then you just let it solidify. When it solidifies, it creates this nice, smooth, glassy, solid surface that colonies will grow on very easily. We can add whatever nutrients we want to that agar. We can put in whatever protein source we want, whatever carbohydrate source we want. We can add minerals to it. We can add vitamins to it. We can add to agar to make it whatever perfect culture media we want. And this is also a really critical thing down here at the bottom. Microbes can't eat agar. They can't destroy agar while they grow on it. Now, I say microbes can't, but there are actually a couple of examples in the scientific literature. There's maybe one or two species of microbe that can degrade agar. They can use it as a food source, but most of them can't. So we don't have to worry that the agar is going to disappear in the incubator because the microbes are eating it. They're not. They're eating the nutrients that we put in it, but they're not eating the agar itself. So it makes a really nice culture material. And all you do in a modern lab is you buy agar as a powdered substance, a granulated substance. You add the water, you heat it up. You can heat it up right in the microwave in the laboratory, melt it, and then you can pour it into petri dishes. Now, you can also skip all that. You can purchase pre-made agar plates. And laboratories that don't go through a lot of agar will often do that. They'll just buy the plates already made. But most microbiology labs make their own plates because it's less expensive to do that. So, in many research laboratories, for example, professors will hire students to make plates, to pour agar plates, because they go through a lot of plates in their research, in their laboratory. Yeah. Okay. Now, the last thing I'll talk about today is what's on this slide. The way we use agar mostly is in plates. And you can see an image up here at the top of the slide of an agar plate. We pour the agar into the bottom of the petri dish, we let it solidify, and then we use this surface to grow cells. It's a nice broad surface. We can get individual colonies growing if we want. We could do quadrant streak plates, we could do spread plates, whatever we want. But there are times when we don't need all of this space. And we can pour agar into test tubes instead. And there are two ways we do this. There's what we call a slant agar tube, and there's what we call a deep agar tube. So again, we're pouring liquid agar into a test tube instead of a plate, and we're letting it solidify. This tube, the way you get it to harden on a slant like this is just by tilting the tube while the agar dries or solidifies. So you get this slanted surface. And the reason you would do this is because you get more surface area if the agar solidifies on a slant like this. So I can take my loop and dip it into a broth culture, and I can streak cells right down that surface. It's just agar, just like a plate. It's just that there's less room in here than there is on a big plate. Now, why would I want to do this? Why would I use a slant instead of a dish? Well, sometimes I don't need to grow colonies. I don't need to do colony morphology. I don't need to count colony forming units. Sometimes I just want to know something about the cells. Sometimes all I need to do is grow a few cells on this agar here because I want to know some specific metabolic ability they have or something. If I don't need to grow colonies at all, I can use this slant tube instead. It has enough surface area for me to spread the cells out a little bit, but it's not enough to grow colonies on. Now, what about this deep? There's very little surface area up at the top of this tube. Remember, this is solid. This is solid agar. So I'm never going to smear cells on top of a deep. Instead, what we use this tube for is if we think we have a microbe that finds oxygen toxic. There are lots of microbes that can't grow in the presence of oxygen. So we have a special tool called an inoculating wire. All that is an inoculating loop where the loop has been snipped off. And all we're left with is the wire. You dip the wire into broth culture, and then you stab it down into this agar. And if you have an organism that can't grow in the presence of oxygen, it can grow way down deep in this tube because there's very little oxygen down here. So we use deep to grow oxygen sensitive organisms. We use slants if we're trying to grow cells here, but we're not trying to count colonies. We're not trying to determine colony morphology. We're just trying to learn something about the metabolic characteristics of that organism. These are just some commercially available slants. It's hard to tell, but they have a slanted surface to the agar right here. And these are some deeps. You can see where somebody stabbed this agar, and the white that you see here, that's that stab line. Over here, this stab line has grown cells. How do I know the difference? Because this one's all cloudy. Can you see that? And the stab line, while it's really thin right here, over here, the line has gotten really thick because cells are growing in here. Cells are growing through out of that stab line and into this deep agar. And they've turned the media that cloudy color. So we don't have to put agar into a plate to use it. We can put it into a test tube and use it as well. And as we go on this semester and we talk about all different kinds of media and we start doing metabolic testing, you'll see slants. There are slants that get used in this course in order to do that kind of metabolic testing. All right, we are out of time. Does anybody have any questions about what we've talked about today? We will continue on with this on Wednesday. Just a quick reminder, don't forget about your exam today. You'll want to get that into me by 8 o'clock tonight. And I think that's all I have for you. All right. If you have any questions or problems, just let me know. Send me a message through Blackboard, and I'm going to let you go. Have a good rest of the day, okay? We'll see you next time.
Thank you too.
Bye-bye.